In this week's lab, you'll be studying respiratory anatomy. So we're going to do a review of respiratory anatomy and their functions, the pathway of air from the external environment to the alveoli where gas exchange happens. And we'll also talk a little bit about Boyle's law and the pressure-volume relationships that allow inhalation and exhalation to happen. These are two of the models that you'll look at this week. This one is a single sagittal head section that lets you visualize the structures that are here in the upper respiratory tract. And then you will also look at other gross anatomical structures in the torso models. So functions of the respiratory system include several things. One of those is that it serves as a pathway for air to get from the external environment to the alveoli where gas exchange will happen. That's really one of the big functions of your respiratory system. So that lets us do pulmonary ventilation, which it has two parts: inhalation, also called inspiration, and exhalation, also called expiration. We do gas exchange via diffusion, and that's between the alveoli and the blood vessels, those pulmonary capillaries. And then later, those pulmonary capillaries have helped to ultimately bring oxygenated blood back to the left atrium and into systemic circulation where gas exchange will happen with the tissues. That gas exchange works along partial pressure gradients, which is, in its simplest form, says that if there's more of a gas in one place than another, the gas will move or move passively, diffuse to the place where that gas has less concentration. Odor detection is important. That's done in the nasal cavity where the olfactory receptors are. Odor detection is important in defense. It's also important in helping us notice things like food that smells off. You'll notice in elderly or people who have anosmia or dysnosmia, potentially even people who are recovering from COVID-19 as well, that having a lesser ability to smell means there's a lesser ability to taste, because taste is a combination. It's about 80% smell, and the detection from taste receptors are in and around the tongue and the epiglottis. So being able to smell something that's off or smell something that's new is a defense for us. You also have defense against antigens from the nasal cavity, really through the epiglottis and part of the trachea. And that's through pseudostratified ciliated columnar epithelium that's in the nose, and it's also in the trachea in the upper respiratory tract. There are goblet cells that make mucus. That mucus has IgAs, which help to keep pathogens from moving through the mucous membranes into the deeper tissues. And then the last function we'll talk about is sound production. That happens at the larynx, where the vocal cords are, and the paranasal sinuses. So those are these open areas, they're kind of like little caves in the skull that allow us to do resonance. So sound resonates or bounces around in those cavities, and then it helps us project the voice. These are the key organs of the respiratory system. So you're going to want to make sure you know the pathway of air from the external environment through the nose, all of these other structures, and ultimately to the alveoli. So make sure you practice that, and we're going to talk about each of these organs and briefly what they do. So it is normal and it is good to breathe through the external nares or the nostril into the nasal cavity. In there, again, is that pseudostratified ciliated columnar epithelium. There's nasal hairs or nose hairs, which are macroscopic hairs. We have the nasal concha These are bony protrusions that go into the nose, and they give more surface area for the epithelium and for warming, moistening, humidifying, and filtering air in the nasal cavity. These grooves between the nasal concha are called the nasal meati, or meatises. So from here, air is going to move into the nasopharynx, and then through the oropharynx, and then into the laryngopharynx. And you can see over here that those separations are delineated. The epithelium here is stratified squamous because, at least for the oro and laryngopharynx, that's also where food is as well. You'll see there's tonsils in here. Tonsils you learned about with the lymphatic system help to capture antigens from the air that we breathe. Air then has to go by the epiglottis through the larynx where the vocal cords are, so that's where we're going to make sound. And that area in the beginning of the trachea are protected by cricoid cartilage, which is hyaline cartilage, and the thyroid cartilage, which is also hyaline cartilage. The epiglottis is elastic cartilage, so if you're eating food, the epiglottis should close to cover the airway. If you're breathing, it should stay open and let air pass into the airway, further into the airway. So you'll see the trachea here has these tracheal rings that go about 3/4, some books say about 2/3 of the way around the trachea. So this is a view of the trachea where we're looking down into it. It's a transverse section. And you can see the tracheal rings here. They help to keep the airway patent, help to keep it open so it doesn't collapse on itself. The posterior aspect of the trachea has the trachealis muscle, and one of the nice things about that is if you intake food through the esophagus and that food happens to be a little bit big and the esophagus has to stretch, you've got this muscle here that allows for that stretch into the trachea a little bit, and you probably kind of feel it as like, ooh, that feels awful going down, or it feels kind of pokey going down. If this was hyaline cartilage, it's possible that food might get stuck in the upper area of the esophagus, and then you're out of luck, because then you can't get energy to be a productive member of your tribe and help the tribe. So once air is moved through the trachea, it reaches this area called the carina, which is especially sensitive to dust and debris, to harsh chemical odors or noxious chemical odors. It's one of the last places where you can try to cough something up before it goes into the lower respiratory tract. At this bifurcation or split, you'll see the right primary bronchus and the left primary bronchus. And if you notice their angles, you can see that the left is much less steep than the right. So if I was a pea or some kind of piece of food, I'm much more likely to go into the right primary bronchus than the left primary bronchus. So from there, the bronchi will continue to split into many secondary bronchi, those are outlined in blue, and then many tertiary bronchi that are outlined in green. Now, lots of these do have specific names. You don't need to know those specific names. You'll see that each lung is composed of lobes, 2 on the left, 3 on the right. Each lobe is also covered by a visceral pleura, much like the visceral pericardium that you looked at, and the chest cavity or thoracic cavity itself is lined by a parietal pleura. So these secrete pleural fluid that helps to reduce friction when the lungs are moving against all the other organs that are in the thoracic cavity, which is a pretty busy place. You can also see here the diaphragm, which is the primary muscle for breathing, along with the external intercostal muscles. then ultimately, each of those tertiary bronchi are going to ultimately end in these respiratory bronchioles and then terminal bronchioles, which carry air to the alveoli. So an alveolar duct is a combination of these small bronchioles and groups of alveoli. Alveoli are composed of simple squamous epithelium, and you can see them here surrounded by the pulmonary capillaries. Keep in mind, this blue vessel is a branch of the pulmonary artery, which has come from the heart, from the right ventricle, and then the red one is a branch of the pulmonary veins. Vein because it's going toward the heart, red because it's carrying oxygenated blood. Ultimately, gas exchange will occur here between the alveoli and the bloodstream. And these other cells that you'll learn about in lecture, for the most part, type 2 alveolar cells are surfactant secreting cells, which is sort of like a non-stick cooking spray for the insides of the alveoli. Helps to keep them from collapsing. And then the purple ones are your old friends, the macrophages, which serve as antigen-presenting cells. So again, another defensive function for the respiratory system. Before you come to lab, please do some reading and review about normal inhalation and the pressure-volume relationships that are involved there. We have a model in the lab that you can look at to visualize those, touch and look at to visualize that. And then muscles of forced inhalation. Please do the same thing for normal and forced exhalation. So you've already learned about these muscles in A&P1. We're just looking at them in a different context. And this is one of the models on which you'll be able to see those muscles. So lastly, let's look at the respiratory histology that you'll be looking at. 2 slides. And again, you've seen both of these slides in A&P1. We're just looking at them from a new perspective, a deeper perspective. So the trachea has, again, pseudostratified ciliated columnar. In this trachea, the goblet cells are a little bit harder to see. They are these sort of open spaces here that produce mucus. Hyaline cartilage is here. This is some adipose tissue that helps to cushion the trachea. The cilia help to move the mucus, and they're in the lumen, so in the inside area. And a trachea's job is simply to be a passageway for air and do a little bit of filtering as well. And then for the lung, we have 2 views of the lung, total magnifications of 40 and 100, so that you can see a little more detail. These are alveoli. So everything that is within the green space here, with the exception of this one circled piece, those are all alveoli. This is the simple squamous epithelium, perfect because it's super thin, allows diffusion to happen rapidly across the alveolus wall to the bloodstream, meaning oxygen and carbon dioxide gas. Those are O2 and CO2. Please write them correctly. They're not this or this. Now this area I've circled in green here, which is also right here. That's a blood vessel. And then the bronchiole, I'll circle in a different color. The bronchiole is here. And remember, it is an organ because it's composed of multiple tissues. And one way you can identify this, I think the easiest way, is to look for this difference in tissue versus the alveolus.